Mannose binding lectin pathway of complement system. The lectin pathway of complement system is also antibody-dependent pathway just like classical pathway. We also have many other similarities of the lectin pathway with classical pathway. The components of lectin pathway include MBL, which is mannose binding lectin. Actually, lectin is a compound which binds to mannose, and mannose is carbohydrate-like compound found in membrane of bacteria. The MBL binds to that mannose of bacterial wall just like in classical pathway. The C1 complex interacts to antigen-antibody complex. Along with MBL, the complex consists of two other proteins, MASP1 and MASP2, namely mannose-binding lectin-associated serine protease 1 and mannose-binding lectin-associated serine protease 2. Here MBL acts like C1q while MASP1 and MASP2 acts like R2 and S2 of classical pathway. All the three components combine and form a complex on antigen surface, also called C1-like complex. A pathogen is when traced, the complement system will encounter it. The mannose-binding lectin binds to mannose in the membrane of pathogen, forming a complex of MBL, MASP1 and MASP2. The complex then acts on complement proteins and activates them one by one. Firstly, it cleaves C4 protein into C4A and C4B. Then it cleaves C2 into C2A and C2B. After that, the C4B and C2B combines together and forms an enzyme complex called C3 convertase, which is actually C4B2B. Hence, the C3 convertase will actually be C4B2B. The C3 convertase works on C3 and cleaves it to C3A and C3B. C3A helps in inflammation, while C3B along with C3 convertase forms C5 convertase. Then C5 convertase will act on C5 protein and will cleave it into C5 and C5b. On the pathogen membrane, the C5b molecule recruits C6 and C7 and gets into membrane of pathogen. So we get C5b C6 C7 complex. Then this complex recruits C8 protein and we get complex in the form of C5b 6 7 8. The complex has capability to polymerize C9 molecule in pathogen membrane. As C5b678 initiates the polymerization of C9 protein to membrane of pathogen, we get poly-C9 in the pathogen membrane, which forms pores in the pathogen membrane and is called MAC. The MAC leads to calcium ions concentration increasing in pathogen, which eventually bursts the pathogen via osmotic lysis.